Okay. I'm attending from Başkent University, Molecular Biology and Genetics Department. My title, Genome-wide Analysis of Gata Transcription Factor Family in Amborella Trichopoda. Genome-wide analysis, firstly I want to say why genome-wide identification and characterization on plants. Genome-wide analysis provides raw data to understand how plants respond at the gene expression level, especially in the stress-related pathways. After characterization and identification, in silico data are obtained to be associated with various stress. These informatic data are very important for future studies and to know the gene and protein family in the related pathways, even if gene expression studies are not carried out at the transcription level. Gata transcription factors are a highly conserved family of proteins that are present in a wide range of organisms, from vertebrates to plants and also fungi. Plants are highly adaptable organisms to their environmental due to their sustainability, unable to lead the conditions that cause stress. This makes their stress response mechanisms largely flexible. The molecular mechanisms behind the phenomenon lies behind the protein that regulated which gene would be expressed, and when the expression would happen, gene expression is regulated strictly by proteins called transcription factors. And plants depends highly on transcription factors' abilities to provide the adaptability they essentially need. Amborella Trichopoda are plants. Amborella Trichopoda is a monotypic plant species from the Amborellaceae family based on molecular studies. It's thought to be the most primitive group of angiosperm plants. Since it is an ancestral species in the evolution of flowering plants, an international working group is working to reveal the genome sequencing. The Amborella genome will shed light on the first stage of genome duplication in the evolutionary past. In addition, role and content of gene exchange between plant mitochondria and cell nucleus and in the evolutionary course of flowering plants can be determined by the Amborella genome. Gata transcription factors. Gata gene family is one of the most conserved families of transcription factors, playing a significant role in different aspects of cellular progress in organisms ranging from fungi to angiosperms. Gata transcription factors are DNA binding proteins having a class 4 zinc finger motif followed by a highly basic region and are known to bind a consensus sequence. Especially in plants, gatas are known to be involved in light-dependent gene regulation and nitrate assimilation. In plants, gata transcription factors have not yet been studied extensively, and knowledge about this class of transcription factors remains elusive. The first plant gata transcription factors, NTL1, identified from tobacco, I think, Spora crassa, and IT2. That functions in nitrogen metabolisms. Recent studies carried out in other plants have revealed involvement gata transcription factors in regulation of various stress-responsive genes. Also, nitrogen metabolisms, flowering, developmental-related genes, and hormone signaling such as gibberellic acid, auxin, stokinin, or something. In this study, it has been identified and characterized gata transcription factor family in Amborella Trichopoda. 20 gata transcription factors have been identified. Their chromosome map, exon intron regions, cis elements, protein patterns, 3D structure, phylogeny, and mRNA analyses were made. As a method, we have 5 sections. First, the identification of gata genes in Amborella Trichopoda. Gata gene sequence were racked from phytosome database using keywords in the search with plant transcription factor database and FAM database. To identify Gata proteins in the Amborella Trichopoda'da genome, both BLAST-P and genome database and hidden Markov model searches were performed against the Amborella genome. Two, Gata proteins of identification of motif patterns, locations, and three-dimensional modeling. Motif presence in the Gata proteins families were identified by using multiple expectation maximization for motif elucidation tool, predicting domains and important states by using interpret scan tool, three-dimensional structures for all the proteins were reported by using FIRE tool, and three, phylogenetic analysis, physical, chromosomal, and location. Phylogenetic analysis was reported by MegaX tool for 20 Gata genes. Gene structure display server program tool was used to estimate the exon intron setup of Gata genes. The location of genes on the chromosomes was determined by using MapGene tool. For examination of these regulatory elements in the promoter region and subcellular localization. 2 kilobase sequence in the upstream region for the 20 Gata genes were analyzed in the plant care program. And the last one, plant small RNA target analysis. PC RNA target was used for the relationship between the transcript of the gene and mRNA targeted for silencing. First, the results. Table contains detailed information on 20 genes identified in Amborella. It looks very complicated, I'm aware. In this table, you can see number of amino acids, molecular weight, instability index, and also cellular localization of Gata, 20 Gata genes in Amborella. These genes generally distributed in the nucleus, cytoplasm, and to laser extent other organelles such as nuclear peroxisome or something. And also most of proteins looks unstable, just one of the stable protein. It's Gata 11. You can see here. In here, we mapped which transcription factors is located on which chromosome. According to results, almost every Gata gene is located on different chromosomes. Only Gata 9 and Gata 10 are together on chromosome 25. This figure showed that 20 Gata genes at exon intron regions. Blue lines represent introns, yellow lines represent exons. Gata 12 has no any introns, but Gata 2, 4, and 9 have 7 introns. In this figure, used to protein motifs pattern as a result of meme-suite base. Compared to domains via interpret-scan data, all motif patterns represent the zinc finger motif patterns. And also you can see here motif consensus sequence. This figure shows 3D models of the 20 Gata proteins. All protein models show 98 to 99% stability. FIRE2 style programs model on protein sequence using the stimulated annealing method. Since Gata proteins are a common family in plants, animals, and fungi, the stability index of conserved protein models is quite high. Gata genes were found to target 2,215 mRNA based on the target mRNA analysis, and also This regulator contains more than 100 regulatory elements related to environmental stress, photosensitive environmental response, or something. I want to say, for example, you can see this photo, maybe Gita box, MAB, myoglobin binding protein. Tata box is important, really, these regulatory elements. And you can see some of them, this photo also. Here we want to show the phylogenetic relationship between Geta proteins. Actually, we could not change the names because we had a problem with presentation time. Just I can say the Geta transcription factor family is a highly conserved family among species, and the resulting phylogenetic tree confirmed this data. Conclusion. In order to first, in silico analysis enables understanding of DNA-protein interactions. Here we made genome-sized in silico analysis of the Amborella and Gata genes, which is the plant species. Although it gives us limited information on the evolutionary process, we were able to obtain important reference data for plant-plant or plant-animal interactions. For in vivo analysis to be performed with Gata genes in the future, data defined at the transcriptional level will be available. In order to determine the efficiency of Gata genes in stress-related pathways, in silico analysis of the relevant genes should be done before qRT-PCR studies. Therefore, this study is a preliminary study to understand transcriptional regulation at the level of gene expression. Besides, this informatic analysis revealed that Gata genes might play an important role in the stress response for Amborella Trichopoda cultures, and suggested that these genes could be used as functional markers for in silico analysis for future study. And thank you for listening. Is there any question? I can say something.